I'm Dr. T and welcome to my office. So today I'd like to talk about controlling your enzymes when you're the cell that has the enzymes. Okay, in a previous video I talked about enzyme inhibition, but that assumed kind of that you are not the cell whose enzyme it was. So let's say you are the cell. You've got a couple of approaches to doing this. If your enzyme's in a pathway, you got a fairly straightforward approach. Just stop the pathway someplace else. But wait, I want to stop the pathway. How do I do that? Or maybe I don't want to stop it. Maybe I want to crank it to 11. How do I do this? Okay, so one of the classic approaches cells are going to do is known as an allosteric site. So you have your enzyme, and in this case it has two binding sites. You've of course got the standard active site where it's going to do its business, but then you have a control site, and you might have multiple of these. Nothing says it's just one. This is where control molecules will bind. If you're thinking, hey, this sounds a lot like a non-competitive inhibitor, yeah, basically, it's the same idea, but we don't really call it that because one, it's a control regulatory system within the cell, and second, it doesn't necessarily have to inhibit. A good number of them don't turn the knob down, they turn it up. So this is called an allosteric site, and so now you have allosteric regulators, things that bind to it. So a couple classic examples would be, if you think about glycolysis, a lot of the enzymes there will have allosteric regulators that are things like AMP. AMP is going to be present. Remember, that's adenosine monophosphate. It's going to be a spent adenosine triphosphate that's had all the energy pulled from it. So AMP or ADP can bind to some of these enzymes, and by binding to them, that's telling the cell, dude, I don't have enough energy. My ADP is being, my wallet is empty. Make it not empty. So in that case, those couple of key enzymes in that pathway are going to get cranked up. They're going to become more active. Whereas they'll also have another site that binds ATP. Hey, my wallet's full. I can wait. I can conserve this. That's going to crank them down. And they're actually balancing how much is bound from one side to the other. And this allows us to create what is effectively an analog chemical computer, which is actually a really interesting thing of thinking about this. Okay, so what else can I do? Another approach I can take is going to be looking at phosphorylation. So I can phosphorylate my enzymes, and I can either turn them on or turn them off. Usually turns them on, but not always. It sometimes turns them off. The enzyme that does the phosphorylation is called a kinase. An enzyme that removes the phosphate group from another enzyme is called a phosphatase. Okay, so now what I can do is I can have something tell my kinases or phosphatases to go do their job and phosphate. It's like, wait, why don't I just communicate directly with my enzyme? Good question. So one approach is that, hey, if I want ATP in the loop, that is one way to get ATP in the loop kind of scenario. Second, though, is if I activate a single kinase, that kinase isn't going to activate one enzyme. It's going to activate a bunch. Not even necessarily the exact same one. So now what I can do is I can take that signal that was very quiet. Maybe I took in a hormone or something. I have a very small signal, and I can amplify it. So once again, we're getting back to this idea of a computer. Now, I can have different things that are activating and deactivating my kinases as well as my phosphatases. It wastes a bit of energy, but it allows me to start making, once again, an analog computer here. Some of them are turning the pathway up, some of them are turning the pathway down. Some of them are activating some pathways and others other pathways. When you see a molecular biology text and it has these weird cascades where it's like, and this enzyme adds a phosphate, let's see if I can talk, to this other enzyme which does this, to this side, and they're like, why this whole mess? That's usually an amplification pathway and or parts where you can have other checkpoints. Speaking of checkpoints, there's another interesting approach going back to those allosteric sites. Let's say I have a pathway. I'm making something. At a certain point, I'll have made enough, so I would like to stop this. So quite often, the final product of your pathway will be an allosteric regulator for one of the early enzymes within your pathway. As I mentioned, ATP interacts with a lot of the major control enzymes. And it's not all of the enzymes. It's usually the ones that are going to have either a branch in the pathway where the pathway can go in a couple directions, or they're a very one-way step, something that uses a lot of energy that would be costly if you don't turn it off. Remember though, ATP is one of my final products from glycolysis, citric acid cycle, etc. And so that is going to be an allosteric regulator that inhibits my enzymes in glycolysis and citric acid cycle. This is what we call feedback inhibition. I'm getting feedback. I'm getting feedback that I've got plenty of ATP and I can cool it. And so I'm going to do so. So when you think about enzyme regulation, one of the key things to think about is that, hey, this is the cell acting like a computer, but at a molecular level and, to make things more interesting, analog. So with that said, have a wonderful day.